This is a slideshow on basic anatomy. And I finished the cytokine stuff yesterday. So for anatomy, if you know the generals, you're okay, pretty much. You don't really need to be an anatomist to be an immunologist, to be honest. So there are some terms you need to know. There's primary or central lymphoid tissue, and then there's secondary and also called peripheral lymphoid tissue. The primary lymphoid tissue is tissue where new immune cells are produced and where lymphocytes develop antigen receptors. So basically, bone marrow and thymus are lymphoid tissue, central lymphoid tissue. Peripheral lymphoid tissue is where lymphocytes aggregate, where adaptive immune responses are mounted. So lymph nodes, the white pulp of the spleen, mucosa-associated lymphoid tissues also. There is such a thing as tertiary lymphoid tissue, and that is temporary structures that form during an infection, usually in close proximity to the infection. You have B cells organizing into follicles that eventually disappear when the infection is fought off. And that is a tertiary lymphoid tissue. Not really covered in this class, though. So MALT is mucosa-associated lymphoid tissues. That's what it stands for. And MALT is a type of peripheral lymphoid tissue. It includes GALT, NALT, and BALT. GALT is gut-associated lymphoid tissues, and that's going to include the tonsils, the adenoids, the appendix, and Peyer's patches in the intestine, mainly small intestine. And then we have the nasal-associated lymphoid tissue and the bronchus-associated lymphoid tissue. And these are all sort of lymph nodes, but they're close to the mucosal surface. So they basically drain the stuff that comes into your system through the mucosal surfaces. Now, lymph is essentially excess interstitial. So it's interstitial fluid that is in excess of what tissue cells have a use for. It is overflow from fluids and the solutes that have leaked out from the blood and tissue cells. Okay, and you do have blood constantly leaking out a little bit of its fluid into interstitial space. That fluid, if it's not drained by the lymphatics, it will accumulate and you'll have swelling build up. So lymph has to move, or else, you'll have swelling. Lymph is what drains the infection. It's where pathogens and their antigens flow. It contains pathogens and their antigens. And it is a constantly moving fluid. Eventually, it returns to the bloodstream after passing through lymph nodes. Okay. The lymphatic system has functions outside of just immunity. They are required for the absorption of dietary long-chain fatty acids. So in the small intestine, bile will emulsify triacylglycerols, cholesterol, and other lipids. They get cleaved into glycerol and fatty acids by pancreatic lipase, and then they're packaged into those big chylomicrons by interaction with the embedded apolipoproteins. And this is how they're absorbed. Apolipoproteins bind to receptors on gastric epithelial cells, and then they are transported into lacteals. So the chylomicron gets transported into lacteals, which are tiny lymphatic vessels that drain little areas in the intestine. From lacteals, chylomicrons are transported through the lymphatic system, and they enter systemic circulation, i.e. the bloodstream, through the thoracic duct. The thoracic duct is where all lymph from the lower left thirds, or I should say 3/4 of the body, end up draining to. So they get dumped into the left subclavian vein. Now the organization of the lymphatic system includes the presence of a central thoracic duct, which is where all lymph is dumped into the circulatory system. So eventually all lymph makes its way back to the blood. It also has one-way vessels, so it does have valves that don't allow fluid to flow backwards. That helps ensure that pathogens are going toward lymph nodes, not swimming away from them. So your fluid can't really flow backward. And that also helps prevent fluid buildup. So this ensures that all constituents of lymph that want to enter the bloodstream from the lymphatics have to first pass through lymph nodes where antigens are processed and surveyed before dumped into either the left subclavian vein through the thoracic duct or into the right subclavian vein by the right lymphatic duct. And here is what the lymphatic system essentially looks like. You do have endothelial cells making up the lymphatic capillaries and the lymphatic vessels. So you do have endothelial cells, but they're a lot more permeable than the vascular endothelial cells. So basically, if it's fluid, it can go in to the lymphatic. Basically, the purpose of the lymphatic endothelium is to just create a separate compartment. It's not really to regulate fluid exchange. So all fluid tissue ends in the lymph, and it flows down a lymphatic vessel where it's collected and drained, and eventually it ends up entering through an afferent lymphatic vessel into a lymph node. So you'll see the little arrow I drew in there. And in the lymph node, you have, of course, lymphocytes. You have B cells in the follicles. You have T cells in the pericortex, and then the medulla, which is where plasma cells are present. And also macrophages are present there to clean up some of the lymph, hopefully thin it out so it doesn't cause any backups. So antigens and antigen presenting cells are going to enter the lymph node through the afferent lymphatics. New T cells and B cells are going to enter the lymph node through high endothelial venules. So they actually enter through the blood. So you do have blood feeding the lymph node. And that is the site where T cells and B cells are going to enter a new lymph node. For example, if the T cell swung by a different lymph node and it didn't find its antigen, it can enter circulation and go to another lymph node where it may find antigen. Or if it's a brand new T cell and it's hoping that it will find an antigen inside a lymph node, then it will also enter the lymph node through the high endothelial venules. The reason they're called high endothelial venules is because the endothelial cells at these HEVs are very tall and narrow. So I guess they're high, so to speak. And now anything that's left over after the lymph nodes clean out some of the debris and all of the antigens in the lymph surveyed, anything that's left over is going to drain from the lymph node out through the efferent lymphatics, which is the outgoing lymphatics. And that will either end up in either that's going to go next to the subclavian vein or it'll go through another lymph node first, and then it will be dumped into circulation through one of the subclavian veins. Okay. So the thoracic duct is the site where about 75% of lymph returns to the circulatory system. So everything in yellow on this person here, any lymph from any of those yellow areas is going to be dumped into the left subclavian vein through this central thoracic duct. And so this ensures that there's only one way for pathogens to possibly make their way into circulation after being introduced into tissues. They're going to have to pass through probably at least one lymph node, if not more than one, before they can ever reach the circulation. Okay. And then we have the right subclavian vein. That's the site where the lymph from the right upper quarter of the body is drained. So the right lymphatic duct dumps the right upper quarter body of the body's lymph into the right subclavian vein. Okay. So essentially that's about it. And then this is a figure showing all of the lymphoid tissues in the body. You've got the thymus, bone marrow, those are primary lymphoid tissues. You've got the appendix, lymph nodes, vessels, adenoids, and lymphatic vessels. And those make up all of your lymphoid tissues. That's pretty much all there is to it. Also, spleen and Peyer's patches in the small intestine are secondary lymphoid tissue. And that is essentially it. Janeway has a really nice figure showing a lymph node. Now, during infection, I should say normally, when there's not an infection, the little areas in the, I should say, the little lobules that make up the lymph node do not contain these yellow circles. Okay, so they don't have follicles. Or they don't have germinal centers. However, when there is an infection, you're going to get a massive expansion of B cells that are proliferating. And proliferating B cells traffic to the same place to undergo division, and that place is called the germinal center. So early on during the infection, a germinal center reaction is going to occur, and you'll have B cells beginning to proliferate after receiving T cell help in the germinal centers. Okay, and normally B cells are normally found, in the follicle, which is this sort of pale yellow area, but the proliferating B cells are found in the actual germinal centers. And as the infection progresses, the germinal center grows larger and larger and larger and larger until eventually the infection is fought off and we no longer need so many active B cells. And that's when they become senile and they begin to shrink. If you were to cut into a lymph node, you would not find them organized like this. You'd probably find here, none here, one here, none here, none here. It doesn't actually go like this. These are supposed to look like sequential steps in the germinal center reaction, starting here and moving this way. And then we have the paracortical area in blue, and that is where T cells are found, and that's also where follicular dendritic cells are going to be found. And T cells that encounter antigen are going to give rise to effector T cells that are going to live. They're going to leave via the vein that drains the lymph node. And then T follicular helper cells are going to localize to the boundary between the germinal center or the follicle and the paracortical area. So they're going to basically be there for a B cell to come to them and present antigen and receive T cell help. And then we have medullary cords. That is where you've got a bunch of plasma cells, usually IgM secreting plasma cells that are going to die pretty quickly. And you also have macrophages here that are surveying the lymph for anything that these lymphocytes might have missed, and scarfing that up and spitting it out after they shred it. And then you have the medullary sinus, which is sort of like the hilum of the lymph node, and then an efferent lymphatic vessel where the remainder of the fluid is able to leave. So if a pathogen wants to get to the blood, it's going to have to go through all of this first, and it happens rather slowly. Lymph is not a very fast-moving substance. It's not being pumped by the heart. Most of what pumps lymph just skeletal muscle tone, and otherwise it's stagnant. It moves very slowly, so those lymphocytes can take their sweet time surveying whatever might be there. And this is just a histological slide showing you the actual cells that are present in the lymph node. So this is what it looks like. And you'll notice that it does start small and get bigger logically. They all sort of have a follicle. And in this one, we have a germinal center. Figure 10.10, activated B cells form germinal centers in lymphoid follicles. So step 1, naive B cells travel to the lymph node through the bloodstream. And if they encounter antigen, they will leave through the efferent lymphatic. B cells that do encounter antigen in the follicle are going to form a primary focus. So a primary focus is a little group of B cells that have encountered antigen and begin making IgM. That IgM is not going to be terribly high in affinity, and it's only going to be made for a few days. Eventually, the primary focus, well, it's made up of cells that are both dividing and producing antibody. And some of those cells are going to move into the follicle to form a germinal center where they can undergo somatic hypermutation. Plasma cells are going to migrate to the medullary cords after they produce antigen, the germinal center to T follicular helper cells, and are told that they can turn into a class-switched plasma cell. They're going to traffic to the medullary cords, and they may leave via the efferent lymphatics. A lot of them end up leaving and entering the blood, and then they're going to migrate, they're going to circulate to the bone marrow. This is especially true for long-lived plasma cells. We do have some plasma cells that don't die after 5 to 7 days. Most plasma cells die after 5 to 7 days. However, there are long-lived plasma cells that can possibly live forever in the bone marrow, so long as they encounter survival signals. And they will be secreting antibody for the rest of your life. So this means that once you encounter a pathogen and begin making antibody to it, you can really think of that as being a perpetual thing. They're going to be making antibody for the rest of your life. At least you're going to have a low amount of it present. So as long as you've been exposed to a pathogen once, you have some antibody to it forever, basically. However, it will decline over time because these are not all going to live forever. Some of them won't encounter enough survival signals and they'll die. What processes are in the germinal centers within follicles? There's a couple of weird things that probably are confusing everybody. There's this thing called somatic hypermutation. Somatic hypermutation is a B cell that has already encountered antigen, presented it to a T cell, is allowed to try to edit its antibody gene to make it higher in affinity. So it's going to randomly edit it, basically throw in random nucleotides and cause mutations. And it's hoping that it's going to make an antibody that is stronger in affinity. Then it's going to go try to find antigen again. Well, if it made a B receptor that is higher in affinity, it's going to be able to find antigen without problem. It's going to take up that antigen, present it again to a T cell, and receive more cytokines and more growth signals and more survival signals from those T cells. On the other hand, because these B cells are editing their antibody genes in a random way, a good number of them, probably most of them, are going to make antibodies that are lower in affinity for the antigen. And those aren't going to be able to bind to antigen as well. So they're not going to be able to take antigen. They're not going to be able to present it to cells. They're not going to be able to receive survival signals. They're going to die. So the B cells that manage to create an antibody that is increased in affinity, basically the stronger you can bind to antigens, the better off you are, and the more likely you are to survive. And this accounts for this thing called affinity maturation. So B cells with high antigen binding affinity will lead to affinity maturation. B cells compete for survival signals and growth factors that are delivered by follicular dendritic cells and follicular helper T cells. They're only going to be able to take up antigen if they have a high affinity for it. There are so many B cells inside the germinal center. They are competing with literally millions of B cells. So when you think about it at that scale, antigen is pretty scarce compared to antigen receptors. Therefore, only the B cells that have the highest affinity are going to be able to capture any of the antigen. And therefore, only the B cells with the highest affinity are going to be allowed to grow and survive and so on, and turn into plasma cells. So over time, the antibody response actually increases in affinity. If you sampled me right now and I just got an infection, you just exposed me to something and you sampled my antibodies right now, you probably wouldn't find any. If you sampled them in a week, you would find low affinity antibodies. If you sampled me in 2 weeks, you would find medium affinity antibodies. And if you sampled me in 3 weeks, you would find really high, really strongly binding antibodies that are very specific at binding to my pathogen causing my infection. So over time, the antibody response increases in affinity. B cell proliferation and differentiation into memory B cells and into plasma cells occurs within the follicles in the germinal center. And class switch also occurs. This is when the constant region of the immunoglobulin is changed to a different constant region. And this is an important thing here. Finally, explain these in a little bit more detail. Follicular helper T cells, TFH cells, they develop during all adaptive immune responses, and they're specialized to provide to B cells, regardless of the defense module that is being promoted during antigen presentation. So regardless of what kind of pathogen it is, you're going to make follicular helper T cells. So TFH cells, those made during type 1, 2, or 3 responses, they're going to play a role in the development of distinct patterns of class switch antibodies. So because you're making TFH cells of a certain kind, you may have a strong IgA response, or a strong IgG response, or a strong IgE response. It is likely that the TFH cell is the principal effector T cell that provides B cell help. So the TH1 cells, the TH2 cells, the TH17 cells, they probably actually don't help that many B cells make antibody. Mostly, the heavy lifting is being done by these TFH cells that are actually located in the lymph nodes where the B cell reaction is happening. The B cell germinal center reaction is happening. When you make a TFH cell, once a T cell specializes and differentiates into a TFH cell, it moves to lymphoid follicles where it remains. And lymphoid follicles, remember, that's the B cell area. Specifically, it's traffic to light zones of germinal centers, and they do not circulate. They are just going to remain there for, as long as they're alive. And like all, I guess this slide really doesn't matter. I guess it matters for B cells, kind of. So they express this protein called ICOS, or the inducible T cell co-stimulator. Inducible co-stimulator. And the ligand for ICOS is called ICOS ligand, and it's expressed on B cells. These proteins are going to bind to each other during antigen presentation. The B cell is going to present antigen to the TFH cell. ICOS ligand on the B cell bind to ICOS on the T cell. This induces activation of both the B cell and the T cell. And specifically for the B cell, it's going to provide a really strong signal for the B cell to go on and become a plasma cell or mutate its antibody genes and undergo the germinal reaction. TFH cells also express the CD40 ligand, which binds CD40 on B cells, and this causes the activation of NF-kappa B in the B cell and increases the expression of B cell survival signals like BCL6, anti-apoptosis protein. So B cells that manage to put an antigen are going to receive this CD40 ligand from T cells and be allowed to survive. So it really does drive survival of the B cell. They are competing literally to survive. Pretty brutal place in the lymph node. So that was my on TFH cells. But follicular dendritic cells are also present in lymph nodes, specifically lymphoid follicles. They are actually non-hematopoietic. They are derived from epithelial cells. But weirdly enough, they are specialized to recruit and activate antigen-specific B cells in follicles. They do also activate T cells, but probably not as much as you'd think. Mostly they're hanging out in the follicles where they're hoping a B cell can come and bind to some antigens on their surface. So they express complement receptors like CR2, and they express C receptors for antibody-coded things. This helps trap complement-coded immune complexes, and an immune complex is an antigen-antibody complex, for the purpose of activating B cells that are specific to the antigen within the immune complex. So basically, you have an infection. The bacteria that's causing infection is producing all these proteins and all these antigens that are draining into your lymphatics and into your lymph node. And they're going to become, those antigens are going to become coated in complement. And instead of just letting all of those complement-coated antigens, swim freely through the lymph node, follicular dendritic cells express complement receptors that bind to the fixed complement molecules that are located on the antigen. And they just hold the antigen basically in place so that a B cell can swing by and bind to it with its B cell receptor, and then take up this little green piece here from the thing that's being held on the dendritic cell. Regular dendritic cells don't only capture antigens for B cells to come and survey. They also produce chemokines that attract B cells to the follicle, so they are largely responsible for the accumulation of B cells within the follicle. And they produce survival signals for B cells, like BAFF, the B cell activation factor, among other things. Remember that the B cells with the highest antigen affinity are going to be bind to these antigens the greatest extent. So they receive the greatest number of survival signals, and that includes from these FDCs and from TFH cells. And that results in affinity maturation. The antibodies that you find in a patient's blood are going to increase affinity over time. They're going to bind more strongly to antigens as time progresses. And that is because they are repeatedly mutating genes that encode their B cell receptor, and then they are repeatedly binding to these antigens on these follicular dendritic cells. And that is also a very helpful thing to help the affinity maturation thing progress. And that is an important role of FDCs. They just hold antigens so that the affinity maturation reaction can happen. So this is a little bit more detailed on affinity maturation, and it looks like a wall of text, but it's probably not very high content. So if you're like gonna scratch your head over this, don't even worry about it. It's probably more relevant for them too anyway. But I want to explain it up front. So B cells mutate antibody genes in the dark zone of the germinal center. So the germinal center has this dark zone composed of a bunch of B cells. And then it has the light zone where you find all the TFH cells and all the follicular dendritic cells. Okay? The B cells, once they mutate their antibody gene, they're going to go see if they've made an antibody that is good at binding to antigen. If they made one really good at binding to antigen, then they're going to leave the dark zone. And once they get to the light zone, they're going to bind to that antigen. They're going to take it up into a vessel and load it onto MHC2. And then they're going to swing by a T follicular helper cell. They're going to present that antigen to the TFH cell. They're going to receive cytokines in response, and they can then re-enter the dark zone and undergo additional mutations. So this process probably lasts about a month or two. And yeah, the antibodies are going to become higher and higher in affinity over time. So it keeps going on because these antigens are just trapped here waiting for a B cell to take them up. So I wrote a paragraph on it, and I'll just read those. So B cells encounter an antigen and receive T cell help. Some then enter the germinal center's dark zone to undergo somatic hypermutation. This is when they mutate their antibody genes intentionally. They induce mutations in their antibody encoding gene. Then they re-enter the light zone of the follicle to re-encounter antigen on FDCs. They take it up and present it to TFH cells. Somatic hypermutation in the dark zone can then occur again. Those T cells are going to provide them with cytokines that say, okay, you can survive, and feel free to mutate genes even further. And by the way, every time it happens, every time T cell help is received, you're going to be making plasma cells as well. So you're going to be making cells that are dumping antibodies with higher and higher affinity as time progresses as well. Now, B cells whose mutations result in a higher BCR antigen affinity compete better for antigens in the light zone, present them to T cells more, receive more T cell help, and proliferate more. And the ones who make low affinity antibodies as a result of mutation are going to die because they can't receive survival signals. As time progresses, clones with higher antigen binding affinity are selected for by those survival signals and those growth factors. This leads to affinity maturation, the observation that the affinity of the antibody response increases over time. So hopefully I have done this its due justice, and it makes a little bit of sense, if not perfect sense. It probably won't make perfect sense for a while. And then this slide, honestly, I don't think it really introduces anything new. The reason I have it is it shows you the location of the TFH cells. So T and B cells are going to meet up at the follicle parafollicular zone border, basically. B cells are presenting antigen to T cells early in the immune response. And then over time, those T cells, later on are differentiated into TFH cells that drive the germinal center reaction. I should say that drive the affinity maturation reaction. And this little picture shows you the overall schematic of, what a follicle with a germinal center looks like. So you have these cells called centroblasts, which are basically rapidly dividing B cells that have just encountered antigen and presented it to T cells and received T cell help. They are going to enter the dark zone where they will proliferate rapidly like dogs, or what is it? They're going to proliferate like rabbit on Viagra. They're going to push all the other B cells out of the dark zone into the light zone. And then they will stop growing eventually. They will re-enter the light zone and try to encounter more antigens on follicular dendritic cells. I've said all I really need to say. So B cells give rise to terminal center B cells that are undergoing the affinity maturation reaction. They're mutating their antibody genes and producing higher and higher affinity clones. They also give rise to plasma cells and memory B cells. The plasma cells are going to move to medullary cords within the lymph node, or they're going to move to bone marrow, especially if they're long-lived plasma cells. Germinal center B cells stay in the germinal center where they continually mutate antibody genes for a month or two. And memory B cells mostly reside in the lymph node or in the spleen. However, they do traffic to tissues, especially inflamed tissues. So you do have memory B cells in your tissues that are ready to go as soon as an infectious agent is reintroduced. Okay. This slide does not show anything new. I don't think I need to say anything there. The spleen is sort of an interesting organ in a way. The spleen is basically a giant lymph node in a sense. It's a little bit more specialized, but it is a giant lymph node. And it is secondary lymphoid tissue. It contains secondary lymphoid tissue. This slide has a whole bunch of pictures, probably more pictures than you could have ever wanted of the spleen. So there's these red pulp areas, and that is mostly what the spleen is made of. It's mostly made of red pulp. These are areas where red blood cells are destroyed by red pulp macrophages that are taking up old senescent red blood cells and destroying them. And then it also has white pulp, which is the actual lymphoid tissue. And white pulp is arranged around central arterioles. And white pulp is composed of follicles and the periarteriolar lymphoid sheath. Periarteriolar lymphoid sheath. That is mainly T cells. It's kind of like the parafollicular zone. It is the site where spleen dendritic cells present antigen to spleen T cells. Follicles are also surrounded the central arterioles, and they consist mainly of B cells, just like you'd see in a lymph node. Secondary follicles also contain a germinal center, and so you can have the germinal center reaction happening in the spleen. And the germinal center will be surrounded by a B cell corona, kind of like the light zone. Each follicle, including this corona, is surrounded by a marginal zone where you have resident non-circulating marginal zone B cells. Marginal zone B cells are a specific kind of B cell that is really good at binding to bloodborne pathogens like Streptococcus pneumoniae and other things that have polysaccharide coats and are likely to enter your bloodstream. They're also good at binding to soluble antigens and immune complexes from the blood, and so on. So I think that's about all I need to say. So they show a transverse section of white pulp and a longitudinal section of white pulp So it looks different based on how you slice into it. But this kind of demonstrates the organization of the white pulp in the spleen around the central arteriole. Now, you're probably wondering, what do I even need about this? The answer is really nothing about the spleen itself, but you're going to want to know about follicles being the site of B cells, the germinal center reaction you probably want to know a little bit about. You want to know T cells are in your parafollicular area. The spleen is lymphoid tissue. It contains lymphoid tissue. Otherwise, the rest of this you don't really need to fuss over. So, kind of move on. And then we're going to look at non-lymphoid tissues that have important immunological functions. So you've got all these barriers that seclude your sterile tissues from your outside world, right? You have the skin, you've got a gut with an epithelium, you've got lungs that basically separate the blood from, the air that you're breathing in. Eyes, the nose, the oral cavity. At all sites, you have epithelial cells joined by tight junctions so that no microbes can get through. In the skin, you have longitudinal flow of air and/or fluids, depending on if you're swimming. Your skin is actually set up so that air flows in a certain way past it. In the gut, you have longitudinal flow of air or fluid, right? You have peristalsis propelling all of the gut contents out of the gut and into the feces. In the lungs, you have the movement of mucus by cilia, and so your lung cells actually contain cilia that sort of scoop mucus out toward your mouth and away from the lungs. Hopefully, that mucus has captured any pathogens that were hoping for a free ride into the alveoli. In the eyes, you have tears, and in the nose and oral cavity, you have cilia that do the same thing, the cilia in the lungs. They basically propel mucus out through the airway, out of the airway, hopefully out of the body, or you may end up swallowing the mucus that you're propelling out of the lungs so that anything that the mucus is captured can be destroyed by the stomach acid. And then you not only have those mechanical mechanisms of preventing infection, you also have chemical mechanisms of preventing infection at these barriers. The skin has fatty acids that actually decrease the pH of the skin and make it pretty inhospitable to a lot of microbes, especially ones that could cause disease. It tends to select for microbes that are not going to cause disease. You have beta defensin, lamellar bodies, and cathelicidin, which are antimicrobial proteins on the skin. And in the gut, your stomach has very low pH. You've got enzymes like trypsin in the stomach and trypsin in the small intestine. In the gut, you've got alpha defensins, you've got reg3 lecticidins and cathelicidin. Those are antimicrobial proteins. You have pulmonary surfactant in the lungs. That helps to capture bacteria and viruses and such in sort of a soapy layer in your lungs. You also have more defense proteins in the lungs, and we will define all of the innate defense proteins. And in the eyes, nose cavity, you've got enzymes in your tears and in your saliva. Lysozyme is going to be capable of cleaving the cell wall of bacteria. And then you've got histatins and defensins hanging out there. And in pretty much all sites, you have a normal microbiota that competes for nutrients and helps prevent infectious agents from establishing themselves as a population, right? So, this is gut-associated lymphoid tissue here. You've got cells. They're microfold cells, right? And they are found in all kinds of mucus-associated lymphoid tissue. The job of a microfold cell is to transport antigens from the mucosal surface into basically a lymphoid structure. So things like bacteria, especially antigens from bacteria, maybe not whole bacteria, but at least their antigens can be transported into lymphoid tissue to be surveyed. You can see how it might be hard to tell pathogenic from non-pathogenic because you're surveying everything in your gut pretty constantly. And so it can be hard to decide whether you want a Th17 response or a Treg response associating with these things. The short answer is if there is no inflammation, then these antigens are going to cause, a Treg response. They're gonna help prevent inflammation against them. On the other hand, if a microbe is causing a problem, then antigens being transported into these lymphoid tissues are going to invoke a Th17 response, and that will cause them to be attacked if they're ever encountered. In addition, Th17 cells and Treg cells have plasticity. When stimulated with certain cytokines, they can actually change back and forth from a Treg cell to a Th17 cell or vice versa. So pretty much all T helper cells demonstrate plasticity. They can change morphology between different subsets. So depending on the context of when the antigen is encountered, if you're looking at an inflammatory context versus a non-inflammatory context, well, in inflammatory conditions, you're probably going to want to mount an adaptive immune response and help fight infection, right? On the other hand, under homeostatic conditions, you'll probably be wanting to make Treg cells so that you don't cause inflammation where it's not needed. So it's useful to be able to switch back and forth because the context in which an antigen is encountered matters a lot. Okay. Here is sort of a look at skin a little bit. A lot of infections initiated by little tiny holes in the skin. Maybe you cut yourself with something, but also we have holes in our skin, pretty commonly. Think of hangnails. Think of even little minor abrasions, or sometimes your skin, it just gets dry, and it may even just crack itself without you realizing. And this can lead to infection. Luckily, you have all of these skin dendritic cells and macrophages waiting to take up pathogens and present them to T cells with co-stimulators. And I can just read the against infection part down here. This is sort of a progression of a skin infection. I think it's from chapter 1. So here is another chapter 1 figure, which I kind of don't like, but whatever. The stratum, the skin is composed of the epidermis, the dermis, and then the what am I trying to say? The cutaneous tissue, right? And the epidermis has 5 layers. Stratum corneum and lucidum are both composed of epithelial cells linked by tight junctions. However, they're not foolproof. Sometimes they flake off, and in that case, you have a watertight lipid layer in the stratum granulosum of the skin. And that is produced by these stratum spinosum cells that are producing lamellar bodies containing lipids and antimicrobial proteins and secreting them into the stratum granulosum. So that any pathogens that happen to get through these first 2 layers of skin and end up down in the granulosum are going to only encounter lipids that make them rather unhappy. Bacteria and pathogens don't really like lipids in general. And then you have the stratum spinosum, another layer of tight junctions. And beneath that, you have the stratum basale, which you start to get into the dermis at this point. This is a list of all the mucosal tissues of the human body, just in case you get them confused. It's sort of self-explanatory. If the tissue makes mucus, it's a mucosal tissue, but that includes some surprising things. The kidneys make mucus and are considered mucosal tissues. They're part of the urogenital tract. The mammary glands are mucosal surfaces, and you can get infections in your mammary glands and cause mastitis. Otherwise, it's kind of not that surprising. You may not know that the eyes are mucosal surfaces, but they are, and they're connected directly to the nose and the nostrils, so it's part of the respiratory tract, very technically. Otherwise, most of this is probably obvious. In the gut, you have goblet cells secreting mucus, and then you have ciliated epithelial cells that are propelling mucus out of the body. Or this isn't in the gut, I'm sorry, this is in the respiratory tract. We're looking at bronchi. So you've got those ciliated epithelial cells propelling mucus out of the lungs and into the mouth where they get swallowed or coughed out or whatever. Maybe you pick your nose and fling it. I guess that's all I really. This is a more detailed look at what lung tissue essentially is. I think there's that much I want to say. The only thing I do want to say is that in the alveoli of the lungs, you have macrophages that help to keep the lungs a sterile place. And the lungs have a very thin membrane of cells separating basically air from blood. And if you have an inflammatory response in the lungs, you have a thickened membrane surrounding, you have a barrier, thickened barrier between the blood and the air, so you impede oxygenation. And then in the gut is a little more interesting. Now, the gut has a whole boatload of microbes that, they may be causing a problem, may not be causing a problem. Goblet cells, they're going to be secreting mucus, and that mucus production will increase under the influence of IL-13, made by Th2 cells and by ILC2s. Surrounding the gut epithelium, you do have macrophages, you do have gut-associated lymphoid tissue and dendritic cells. And then you also have in the gut these things called crypts. We're actually looking at the small intestine only here for crypts. You have crypts in your small intestine. Crypts are little places where, I guess, you usually fold cell down here, but you also have these cells called Paneth cells. And Paneth cells secrete antimicrobial proteins to help reduce the density of microbes right at the epithelium. So hopefully they are kept low in concentration. With that being said, still have a really large number of microbes in the gut of all kinds. It's healthy. So that's about it for anatomy, and I think I'll save antibodies for next time.